Hello, my name's Emily and I'd like to take you through gene regulation data in Ensembl. We'll look at gene regulation data in a region, in a gene, and at the regulatory features themselves. But first I'd like to explain a bit about how our build works and where our data comes from. If you'd like to jump straight to accessing the data, it starts at this time. We use data from a variety of sources. At the time this video was made, we had data in human from ENCODE and roadmap epigenomics, and in mouse from ENCODE. We plan to bring in data from Blueprint and HipSki for human soon, and in future may have many more data sources. All of these projects aim to map large-scale regulatory data, such as histone modifications, transcription factor binding, and open chromatin, in a number of different cell types or tissues. Our documentation lists all of the data we have and where it comes from. Visit each project's webpage to find out more about them. Most of the raw data we have is from ChIP-seq, but we also have data from DNase 1 sensitivity and bisulfite sequencing. We also have all of these data available as raw reads, but we process them to produce what we call features and segmentation. Firstly, we call peaks on all of the reads in each cell line. We then use the signals to segment the genome in each cell line. When we do this, we're looking for patterns in the signal, finding which signals tend to co-localize with each other. We can then categorize these co-localizations into segments with particular regulatory functions, such as promoter or enhancer activity. We do this by comparing to regions of known function. So if we have a known promoter and it has particular histone modifications, where we see those same histone modifications elsewhere, we will identify that region as a promoter. Note that when we call a regulatory feature, that region has not been assayed for that function. It just has biochemical signals that are associated with that function. We then compare all segments in the cell lines and look for patterns. We use 200 base pair bins and check what each of the cells are doing at that position. From this, we create our multi-cell build. This shows us all of the activity that has been seen in all cell lines. That is, it tells us what each region of the genome does. These are called multi-cell regulatory features. From this, we can go back to each of our cell lines and ask, does this activity occur in this cell line? These give the regulatory build for each cell line. These are also called cell line regulatory features. Remember that these functions haven't been biologically assayed and are just predictions based on the ChIP-seq data. So that's the data. How do I get to it? Let's start by looking at a region. I'm going to go to the region on human chromosome 19, which contains the ZNF557 gene. I'll jump straight to the region by selecting human and pasting in my coordinates and clicking go. We're now at the region in detail page. If you're not already familiar with this page, you might want to look at our other help video. Let's look at the detailed view at the bottom. The multi-cell regulatory features are shown by default. I can find out what they are by clicking on them. This pinkish feature is a promoter flanking region. There's also a legend at the bottom, telling me what the colour codes mean. These black lines are transcription factor binding motifs. If I click on them, the motif is highlighted in the pop-up. There's an Ensembl ID here. This is the ID for the regulatory features, in the form ENSR, followed by 11 digits. We could click on this to go to the feature, but we'll look at that later. I can look at cell-specific data here in the region in detail view. To do this, I click on the blue Configure this page button. Here's the regulatory menu on the left. Let's open regulatory features. Here I can see the regulatory build for each cell line and the segmentation. Let's turn on the data for one particular cell line, GM12878, both features and segmentation. We'll also turn on the raw chipset data. This is the raw data that was used to construct the segments and features. This is found in two menus: open chromatin and transcription factor binding sites, and histones and polymerases. We'll start with open chromatin and transcription factors. This is our regulatory matrix. It allows you to add these data more easily. We have transcription factors down the side and cell lines along the top. Where they intersect, we can turn on the track. Click on the box to turn it blue and turn the track on. When I turn on the track, it gives me a track style for that cell line. Some features are already on by default, but they don't actually turn on until I choose a track style for them. As well as turning on tracks one at a time, I can turn on all cell lines for one feature, or I can turn on all features for a cell line. I'm going to turn on all tracks for GM12878 and change the track style to both. This will display the raw chipset reads and the peaks we calculated for them. I'm also going to turn on all histones and polymerases tracks for GM12878. I'd also like to add DNA methylation data. I can do this under DNA methylation. Here's GM12878 whole genome bisulfite sequencing. Now I've added all the data I want, I can close the menu. I can see regulatory features for GM12878. See how they exactly correspond to the multi-cell features. If they're switched on, the colours match. If they're off, they're coloured in grey. If I click on these, I get a similar pop-up to the multi-cell. It has the same Ensembl ID, but it also lists the attributes. These are the pieces of evidence that we use to construct the feature. If we look at our peaks for our transcription factors and histones, we can see that the peaks within the features are listed here. Here's the segmentation. Instead of blocks like the features, the segmentation is a single continuous track. Most of the colours match up between the features and the segmentation. However, regions will differ because the features are a consensus between cell lines. We can see the peaks for the histones and the transcription factors here. Black lines in the peak indicate the motif for that transcription factor. I can get a legend for the signals here. DNA methylation is shown at the bottom. Yellow indicates low methylation, such as at this active promoter. That's how we can find regulatory features for a region. Now let's look at a gene. I'll click on one of the transcripts for ZNF557, then click on the gene ID to go to the Gene tab. Regulation is here in the left-hand menu. Here we can see the regulatory features and segments surrounding this gene for all cell lines. They look very much like the features for the region. In all displays, features, segmentation and raw data are displayed in the same style. This promoter flanking region is active in all of the cell lines studied. This feature is at the 5' end of the gene, so it's reasonable to infer that this is the promoter of the gene. However, this activity has not been biologically assayed. At the bottom of the page, the locations and sequences of all the features are shown in this table. I want to find out more about this promoter flanking region, so I'll click on its ID in the table. I could also have gotten here by clicking on the feature block in either the Region in Detail view or Gene view. If I knew the ID, I could search for it in any of the Ensembl search boxes. This is the summary page, which shows the activity of the feature in different cell lines. There's also a summary of how many of the cell lines it's active in. We currently have 18 cell lines, so it's easy to see graphically, but when we have more, this summary will be very useful. These buttons show the pages you can look at in the Regulation tab. I'll click on Details by Cell Line, which allows me to view the evidence in each cell line. Here we can see the features and segmentation by cell line and a subset of the evidence peaks. If I want to change the evidence I can see, I can click on Select Evidence here. The blue highlighted evidence is switched on and the grey evidence is switched off. I can turn evidence on or off by clicking on it, or I can choose to turn it all on or all off with these buttons at the top. I'll close this menu. I'm currently displaying all cell lines, but I can change this with Select Cells. Now I can choose my cells of interest from the list, or turn them all on or all off. I'll turn them all off and just pick 2 cells. I'm currently only seeing peaks of evidence. If I want to see signal, I can click here. I can also turn the peaks and the signal off from here. So that's how you can see Ensembl regulatory data in the browser. You can also access it via our Perl API. Take a look at our online course to learn more. If you're interested in further regulatory data, you can have a look at our Track Hub. You can get the Data Hubs list from our homepage. This lists all the hubs available, including hubs from ENCODE, Blueprint and Roadmap. Some of them only work on old versions of the genome assembly, so the assembly is listed on the right. Click on the assembly to add the hub and jump to the old assembly. You are now in the hubs page and can configure them from here. Thanks for watching the video. If you have any trouble accessing these data, or if there's anything you don't understand, just drop us a line. We're helpdesk@ensembl.org.